Hey everybody, welcome to another lecture in our Chem 150 adventure. This one is on amino acids. So, all of the proteins on the face of the earth are made up of the same 20 amino acids, linked together in long chains called polypeptides. Now, amino acids are the building blocks for the vast majority of the vast assortment of proteins that are found in all living cells. So this is a diagram of an amino acid. All amino acids have the same basic structure. At the center of each amino acid is a carbon called the alpha carbon. There. The alpha carbon has four groups. It has a hydrogen. It has a alpha carboxyl group. It has the alpha amine group and an R group, sometimes referred to as side chains. The alpha carbon, carboxyl, and amino groups are common to all amino acids, so the R group is the only unique feature to each individual amino acid. A minor exception to this is the structure of proline, which at the end of the R group is actually attached to the amine nitrogen. So, with the exception of glycine, which also has an R group that's just a hydrogen atom, all of the amino acids in proteins have four different groups attached to them, and consequently can exist as either L or D forms, if you remember your stereoisomers, or your enantiomers, or your just chirality, carbon chirality. With only minor exceptions, every amino acid in cells and in proteins is the L configuration and not the D configuration. There are 22 amino acids that are found in proteins. 20 of these are specified by a universal genetic code. The others, selenocysteine and pyrrolysine, use tRNAs that are able to base pair with stop codons in the mRNA during translation. We'll get to transcription and translation in a later chapter. But when this happens, these unusual amino acids can get incorporated into proteins. Enzymes containing these selenocysteines, for example, can include glutathione peroxidases, tetraiodothyronine, 5' deiodinases, and so on. Selenophosphate synthase. There's a lot of examples of enzymes that need these unusual amino acids, like selenocysteine. Pyrrolysine containing proteins are much rarer and are usually only found in archaea. But with beyond the 20 kind of universal amino acids, we'll see that there's also some modifications that amino acids can get that can make somewhat new or different, let's say, amino acids. But there are 20 that are coded for in our DNA. Nutritionalists will divide amino acids into two groups: essential amino acids, they must be in our diet because our cells can't make them, and non-essential amino acids, which can be made by our cells. The classification has nothing to do with the structure, or very little to do with the structure of amino acids. It's just that some can be made by us and some can't. Essential amino acids vary considerably, though, from one organism to another, even in humans, depending on whether they're adults or children. Children are able to synthesize certain essential amino acids that adults are not able to. And so you can see this table here sort of designates the essentials versus the non-essentials. Essentials being, again, the ones we need from our food. So some amino acids that are normally non-essential may need to be obtained from the diet in certain special cases, such as if we have a genetic disorder or if we have a problem making one of our own non-essentials. Non-essential for everybody else, but becomes essential for you. Individuals who not synthesize enough amounts of arginine, for example, or cysteine, or glutamine, or proline, due to illness, may need dietary supplements. There are also non-protein amino acids. There's alpha amino acids that are found in cells that are not incorporated into proteins. These include ornithine and citrulline, to name a few. Both of these compounds are intermediates in the urea cycle, which we probably won't get to over the summer. Ornithine is a metabolic precursor of arginine, and citrulline can be produced by the breakdown of arginine. They're amino acids because they have the carboxyl group, they have the amino group. They're just non-protein. Citrulline is a metabolic byproduct, sometimes used as a dietary supplement to reduce muscle fatigue, if you've heard of it before. So let's talk about some R group chemistry. We separate the amino acids into categories based on their the chemistry of their R groups. So if you compare groupings of amino acids in different textbooks, you'll see different names for the categories, and sometimes the same amino acid being categorized differently by different authors. So indeed, like tyrosine gets categorized as an aromatic amino acid, but also as a hydroxyl amino acid. It's useful to classify amino acids by their R groups because these side chains that give each amino acid its characteristic property. But amino acids with chemically similar side groups can be expected to function in similar ways. For example, during protein folding. So we're going to talk about the non-polar amino acids first. You can kind of see here in this table some of the ways that we're going to classify. So we'll go non-polar, carboxyl, amine, aromatic, hydroxyl, and talk about some of the others also. Okay, alanine. Let's see, do I have a better... Oh, this is one of those tables I was alluding to earlier. So here are amino acids. There's short code abbreviations, single letter. There are 3-letter abbreviations. These are for ease of writing out long peptide sequences. Side chains, you can see here some of those differences, whether or not they're hydrophobic, in X there, what their pKa's are. So pKa's being of the acidic part of these molecules. So aspartic acid, cysteine, glutamic acid, right? These guys right here. They all have an acidic hydrogen that they can lose. So if they don't have acidic hydrogens, then they're not shown with any numbers there. You can see whether they're polar, their essential pH, small, tiny. I'm not really sure what small or tiny refer to here. Okay, so we're going to go and talk about some of the non-polars first. So alanine, ALA, or A single-digit code, is one of the most abundant amino acids found in proteins, ranking second only to leucine in occurrence. So leucine shows up a little bit more. A D form of the amino acid is also found in bacterial cell walls. Alanine's non-essential. We can synthesize it from pyruvate. We'll see that in the citric acid cycle, though we won't look at anabolism. We'll be focusing on catabolism or breakdown. And it's coded for by GCU, GCC, GCA, and GCG. 4 different codons will actually code for alanine. Glycine. Glycine is the amino acid with the shortest side chain, having an R group with only a hydrogen on it. As a result, glycine is the only amino acid that is not chiral. It is its small side chain allows it to fit readily into both hydrophobic and hydrophilic environments. So that's glycine. Here, I'll give you... They're in your book. I don't need to write out the codes. I was going to give you more triplet codes, but we'll just save those for when we're talking about translation. Okay, isoleucine. A little out of order there, sorry. Where's my... proline is the only amino acid found in proteins with an R group that joins its own alpha amino group. So it's a secondary amine and a ring. Proline is a non-essential amino acids coded for by 4 different codons. It's the least flexible of the protein amino acids, and thus it usually gives some sort of conformational rigidity whenever it's present. Proline's presence in proteins does affect its secondary structure. We're going to look at alpha helices and beta strands as secondary structures when we talk about protein structure in next week, or not next week, sorry, later video tomorrow. It's a disruptor of alpha helices and beta strands. Proline is often hydroxylated in collagen, we see as hydroxyproline. And this has the effect of increasing the protein's conformational stability. Protein proline hydroxylation serves as a sensor of oxygen levels and targets HIF, which is a hypoxia-inducible factor for destruction when oxygen is plentiful. We have a system in our body that helps, maintain oxygen levels. Excuse me, and senses when they get low or when they get high. So moving on, isoleucine. Isoleucine is an essential amino acid. It only has 3 codons that code for it. It's got a hydrophobic side chain, it's chiral, and its side chain as well. Leucine is a branched chain amino acid that is hydrophobic and essential. Leucine is one of the only dietary amino acids reported to directly stimulate protein synthesis in the muscle. But there are a few conflicting studies on that, so don't just go start mainlining leucine. Leucine also has a toxicity, and it leads to some what they call the 4Ds: diarrhea, dermatitis, dementia, and death. So there is a problem with having too much leucine. Leucine, though, is coded for by 6 different codons in our genetic code. So again, it's one of the most common amino acids that we see. Valine, another essential nonpolar amino acid synthesized in plants. We can't synthesize it, right? That's why we get it from our food. We see it in hemoglobin when it replaces glutamic acid at position number 6. It actually causes hemoglobin to aggregate abnormally under low oxygen conditions, which causes sickle cell. So we'll look at sickle cell anemia later, how it's a single amino acid substitution that causes this disease, but that's what it's kind of most known for. I mean, if we're going to say anything fancy about valine, it's that, hey, when we put a valine in, instead of a glutamic acid, we end up with a sickle cell. Other than that, again, it's essential and it's nonpolar. And then methionine. Methionine is an essential amino acid. One of 2 sulfur-containing amino acids. Cystine's the other one. Methionine's nonpolar and encoded solely by AUG. It's got 1 codon. This is usually the initiator codon. So most protein synthesis starts with the incorporation of a methionine, and then it's later removed. In prokaryote cells, it's the first methionine is often formulated. So FMET is how it's abbreviated. Formulated methionine. All right, carboxyl amino acids. So there's 2. There's aspartic acid. I didn't give you the 3-letter codes for these guys. Okay, you can always look up. I don't mean the codons. I mean the short letter codes. So we'll just jump back real quick. So actually, those are all here. So if you were interested in those, the tests will be largely, open notes for this summer course. So I'm not too worried about you guys memorizing these. Okay, aspartic acid, non-essential amino acid. It's got a carboxyl group in its R group, readily produced by the transamination of oxaloacetate in terms of its synthesis. And it's nice to talk about that here because we probably won't get to protein anabolism. This has a pKa of about 3.9. So aspartic acid's side chain is usually negatively charged at around physiological pH. That's going to have an important feature when we get to tertiary structures of proteins. So it's got a negative charge on it. Aspartic acid is specified in the genetic code by 2 codons. Glutamic acid, also encoded by 2 codons, non-essential amino acid, readily made again by transamination of alpha-ketoglutarate, another citric acid cycle intermediate, just like oxaloacetate. So made from breakdown of sugar products, basically, we can make these. So it's non-essential. It's also a neurotransmitter, and it has an R group with a carboxyl that ionizes at about 4.1, so the pKa for that one. So it's also charged at physiological pH. Now we have some amino or amine amino acids. So if we had to call them acidic and basic, we have 2 acidic amino acids and 3 basic amino acids. So the first is arginine. Arginine is in some cases essential, but not essential in others. So premature infants can't synthesize arginine, but most people can. synthesize it. So they don't need to take. So it's something you grow into as you get older. You can synthesize it for yourself. Um, let's see. Arginine side chain contains a complex guanidium group. You can see it there. It's got an amino NH2 that's going to have an extra hydrogen on it. So it's usually charged at physiological pH. This guanidium group has a pKa over 12, so it's got a positive charge at regular pH. So this is going to be important. Those positive charges will, form bonds with negative charges within protein structures and help hold them together. This has 6 codons that codes for it. Arginine. Histidine, the only one of the amino acids here to contain an imidazole ring. This is the imidazole functional group right here. It's an essential amino acid in humans and other mammals. Let's see, what can we say about it? Its side chain pKa is about 6, so it has a charge with a slight low pH, but if it's above too high above 7, it might lose it. So again, its pKa is close there to 6. Protonation of the ring results in the 2 NH structures, which can be drawn into 2 equally important resonance structures. So there's this double bond here can move back and forth. Okay, lysine, an essential amino acid encoded by 2 codons. Its R group readily ionizes with a charge of +1 around physiological pH. It can be post-translationally modified to a few different versions of lysine: acetyllysine, hydroxylysine, methyllysine. It can also be ubiquinated, sumoylated, metylated, biotinylated, carboxylated, pupillated, and so on. Orthoglycoxylation of hydroxylysine is used to flag proteins for export from the cell. So there's certain functions for these variations of lysine. Hydroxylysine, of course, just having a hydroxyl group attached to it somewhere. I'm not exactly sure at the moment which of those carbons gets it. Lysine is often added to animal feed too, because it's a limiting amino acid and necessary for optimizing the growth of pigs and chickens. So if they don't have enough lysine, they tend not to get very big. And so by providing them with enough lysine, we ensure that they get as big as they can. Excuse me. We've got 3 aromatic amino acids. Phenylalanine, nonpolar, coded by 2 codons. Metabolic precursor of tyrosine, so we can actually make tyrosine from phenylalanine. Excuse me, an inability to metabolize phenylalanine actually arises from the genetic disorder known as phenylketonuria. So if you have that, then you are unable to break down or to use phenylalanine for any source of energy. Of course, remember, you eat proteins, and so you digest amino acids, and a lot of them get broken down for energy. Some of them get recycled, but your body is really good at just making new amino acids anyways. Phenylalanine is also a component of the aspartame artificial sweetener, if you ever look on the ingredients. Tryptophan, an essential amino acid containing an indole functional group. Metabolic precursor of serotonin, niacin, and then in plants it makes the auxin phytohormone. Serotonin, of course, in our bodies being a neurotransmitter, so we need tryptophan. Of course, they also say that if you get enough tryptophan on Thanksgiving, right, from turkey, that it helps you sleep. People have thought that tryptophan somehow is related to sleep. There's no real evidence suggesting this. It makes more sense to think, though, if you just stuff your face, at Thanksgiving or Christmas or whenever, that you're going to be tired because you're digesting that food. So it's not really that turkey, if you've heard that turkey makes you sleepy because it contains a lot of tryptophan. It's just not true. We'll talk about tyrosine here. It's also a hydroxyl amino acid, so we'll just ignore it when we get to the hydroxyl amino acids, but it does have one. Tyrosine, non-essential. It's got 2 codons that code for it. It's a target of phosphorylation in proteins by tyrosine protein kinases and plays a role in signaling processes. tyrosine hydroxylase actually converts tyrosine to L-DOPA, and so in cells in the brain called dopaminergic cells, tyrosine hydroxylase, an enzyme, will turn tyrosine into L-DOPA, which is an immediate precursor to dopamine, and then it gets turned into dopamine after that. Dopamine, in turn, is a precursor of norepinephrine and epinephrine, so of course you need tyrosine if you want any of those. It's also a precursor of the thyroid hormones and of melanin. So tyrosine, though not essential, is essential, right? We need it for everything. All right, the hydroxyl amino acids, there's a few. We've got serine. This one has, let's see, how many was it? 6 different codons that'll code for it. This one can hydrogen bond with water, so it's polar amino acid. It's not essential for us. We can make it. It's a precursor of many important cellular compounds, including purines and pyrimidines, which are our nitrogenous bases for our DNA, sphingolipids, folate, I believe is a B vitamin, and some of the amino acids, glycine, cysteine, and tryptophan. So it's a precursor in all of those. Hydroxyl group on serine in proteins is a target for phosphorylation by certain protein kinases. Kinases are molecules that transfer or so add or remove phosphate groups, and this has the effect of, we'll see later on in enzyme activity or in protein behavior, that you can turn enzymes on or off, usually modulate their activity with phosphorylation via these kinases, and these hydroxyl groups are a good site for that. Serine is also part of a catalytic triad of what we call serine proteases. And we're going to look at some different types of proteases when we get to enzymes, and so I'll save the discussion for that, but it is one of the serine proteases. All right, and then we're going to skip tyrosine. Since we talked about it, we'll talk about threonine. So threonine is a polar amino acid. This one is essential. We need to get it from our diet. One of 3 amino acids that has a hydroxyl group. Serine and tyrosine are the others. So as such, it's a target for the phosphorylation from proteins. It forms these phosphoester bonds. It also is a target for, I can never say the word right, orthoglycosylation. There's an O dash, I'll write it out for you. O-glycosylation. I never remember if O is ortho or O is just an oxygen. But this glycosylation of proteins targets these hydroxyls also. Threonine proteases use hydroxyl groups of the amino acid in their catalysis, just like the serine proteases do. And they are precursors in, threonine, is a precursor in one of the biosynthetic pathways for making glycine. I think in some applications it's also used as like a prodrug to increase brain glycine levels. Yeah. All right. So that is the amino acids. Now, in terms of talking about their ionizing groups, so the different pKa values for amino acid side chains are very dependent upon the chemical environment in which they're present. So, for example, the R group carboxyl found in aspartic acid has a pKa value of 3.9 when in free solution, but can be as high as 14 when in certain environments inside of a protein. Though that's unusual and extreme, but the different environments affect the different pKa values. So each amino acid has at least one ionizable amine group and one ionizable carboxyl group. When these are bound in a peptide bond, they no longer ionize. So only the N terminus and the C terminus. Let me see. So on the far end of our peptide, I don't think I have a picture of a peptide to show us. Actually, polypeptide. Here we go. No. I guess you could kind of see it here. If this was a long peptide, this N and this C can no longer ionize, but this N can, and this C can. We call this N here the N terminus, and this one over here the C terminus. Sorry. Some, but not all, of the amino acids have R groups that can ionize. The charge of the protein then arises from the charges of those alpha amine groups, the alpha carboxyl groups, and the sum of the charges on the ionized R groups. And so titration or ionization of aspartic acid, which I've got here, you can actually see all those different changes. Now we looked at one of these earlier in the last chapter on acid, the base, and buffers, but here you can see that the amino acid groups are changing. The first group that changes is one of our alpha carbons is going to, or our alpha carboxyl will ionize. And then the next thing that happens is our R group's carboxyl ionizes, and then our alpha amino group loses its charge. All right. So basically, when something ionizes or deionizes within the protein structure, this can have a significant effect on the overall conformation of the protein and on the enzyme, if it's an enzyme, and on that enzyme activity, because it's no longer has the right charges which are intended to hold the molecule together. Now, most proteins have relatively narrow ranges of optimal activity that typically correspond to the environments in which they're found, and that I think I had on this slide here, possibly. Let's see, is this 211? No, that's not 211. So in your book, no, I guess I don't have the picture I want. I apologize. So they have narrow ranges in which they can be found. It's worth noting that the formation of peptide bonds between amino acids actually removes those ionizable hydrogens from both the sides. So you only care about the amino terminus, N terminus, or the carboxyl terminus, or the R groups. And then I guess if there's other functional groups such as sulfates or phosphates, those additionally would have charges on them as well. When we build polypeptides, we're going to link amino acids together. So although amino acids do serve other functions in cells, their most important role is just as constituents of proteins. So these are polymers of amino acids. So amino acids link together, each by a peptide bond in which the carboxyl group of one amino acid is joined by the amino group of the next. They lose a molecule of water, and we call that bond, that special kind of amide bond, we call it a peptide bond. Additionally, additional amino acids are added the same way, forming new peptide bonds between the free carboxyl side and the growing chain and the incoming amino group of the next amino acid. So a chain made up of just a few amino acids is called an oligopeptide, oligo meaning few, where a typical protein is made up of many amino acids called a polypeptide, poly being many. The end of the peptide is with the free amino group side is called the N terminus, and the other side, the growing side, is the C terminus. So we always read a protein from N to C. Okay, so function is dependent on structure, and the string of amino acids has to fold into a specific 3D shape or a conformation in order to make a functional protein or an enzyme or whatever, a structural protein. And so the folding of polypeptides into their functional forms is essentially the next topic that we're going to go over. So stay tuned. All right, guys. See you next time.